Okay, molecules in motion, smaller orbits with high momentum. So I love this photo of Rosemary, a gorgeous girl. And one of the things that always makes me laugh is that whenever Rosemary is asked, how long have you been at Monash, there's this pause and then single word, forever, forever. So I always like to think about that term. It's obviously not forever, sadly for me, but quite a long time, and a beautiful photo. Okay, so I thought I'd just give you a little bit of background where my field is at the moment. So the FDA, the Food and Drug Administration, release this kind of information every year to tell us what kind of new chemical entities or new drugs have been released onto the market. And just to sort of put it into perspective, last year there were 55, which is the second largest in about a decade. So not many go onto the market. And about 60% of them are these small molecule things that are rich in functionality so that they can engage with receptors, and they're small enough to get across membranes and to interact with receptors, where they do the cascade of biology that leads to a physiological response. But just very recently in Chemical and Engineering News was an article, Macrocyclic Peptides Aiming for the Perfect Fit, like a Tetris kind of thing. The hunt for drugs that are bigger enough to interact with proteins, but small enough to negotiate cell membranes is coming full circle. And this was like music to my ears because I've been working in this space for nearly 2 decades. So it's like, it seems that the field has sort of caught up a little bit. And what we're talking about here with these macrocyclic peptides are ones like the ones on the top here. So they're small enough not to be considered proteins like big viruses and things like heparin, which are about 40% of the new chemical entities that are released, these small molecules. And this is sort of a space that is largely held by radiopharmaceuticals. So they're used to image tumours and to treat tumours. They're just the right size. But it seems that the rest of pharma are just starting to catch up. And the reason why they're interesting is that it's all about the shape of the molecule and how they curve and how they move. The motion is really, really important. Because what you'll see, perhaps in some of these slides, is it's the dance between the host or the receptor that these things have to engage with. Don't ever think of them as being static entities. They're both moving. It's like a tango and a love affair between the two. So when I think about macrocyclic molecules and how nature thinks of them, largely what she likes to do is to take 2 cysteine residues and oxidise them together. And why is that generally done? Well, it's because that protects the backbone from being chewed up by proteases. So just that simple act of cyclising the backbone will keep it in circulation a lot longer. And so back in Rosemary's day, she would have had this textbook. Looks like this. You can see, perhaps not at the back, but cysteine's in here, and you had to learn the structure of cysteine. And then when it gets oxidised, you get the disulfide, the sulfur-sulfur atoms together, and this is called cysteine rather than cysteine. And in chemistry, you'll be pleased to know that we don't deliver this kind of chemistry in this way anymore.
Oh, should do it again.
No, we take a photo of it and we put it into PowerPoint. So nothing changes. But disulfides are much more interesting than you would tend to think from that textbook. And at the top, disulfides in peptides can be structural, so they can just simply be the architecture that keeps something in the right shape to interact with a receptor. They can be redox active, so they can reduce at a receptor site and open up and consequently oxidise something else. They can chelate to metals, and they can be allosteric, and that's a term that we use that says that the molecule will change its shape, particularly through the disulfide bridge, and bring the rest of the architecture with it when it wants to interact with a receptor. So allosteric disulfide bridges. And that's important because for 50 years previous, Largely X-ray crystallography, these snapshots of molecules like the insulin molecule, loads of Nobel Prizes have gone towards people doing insulin work. That snapshot is not representative of the bioactive form of that hormone, nor many others at all. So it's a snapshot, a frozen snapshot. And then if you use complicated NMR, where you take the molecule and put it into solution, sure, it can tumble around and it can move, but also all of those conformations that you see in solution are not necessarily representative of the bioactive conformation of the molecules either. It's the dance that happens at the receptor that's really critically important, and we can't see that generally with those kinds of techniques. So why is it important? Well, disulfide containing molecules, think of things like insulin, IGF, oxytocin, somatostatin, all of these so-called undruggable drugs do quite well keeping us going. When pharma says they're undruggable, they're saying we can't make them years in advance, we can't put them on the shelf because they'll degrade, we can't deliver them to you in a pill. That's what undruggable is really meaning. These molecules are super potent, super selective, and they do just fine. Okay. Further to that, if you can think of all of those sulfur-containing molecules that we think of, Australia is super rich in venomous creatures, from things like cone snails, beautiful shells. People pick them up, they pop them in their wetsuit, and then they get stung, and that can stop respiration. You can die from a cone snail bite. Spiders, snakes, anemones, scorpions, all of these organisms make these beautiful three-dimensional peptide structures that are constrained in their movement by disulfide linkages. So something as simple as a toxin out of a scorpion, two interlocked disulfide bridges, going all the way to things like snakes and funnel web venom, where the disulfide connectivity, up to 8 disulfide bridges all knotting and curling around each other to form these three-dimensional structures that nest into the receptor. So they are exquisitely selective and potent molecules. And obviously these organisms don't use them to reduce blood pressure or reduce heart rate or be diuretics or cure pain. They're using them to envenomate their prey, to immobilise things, to eat things, etc. So, but we can learn a lot about these venoms, and we can modify the structures to make therapeutic molecules. They're a very important supply of structural information. So, before I go on to that slide, so in my group, we take that metabolically unstable disulfide bridge, this part of the molecule up here, and we replace it with a carbon-based bridge, which will mean that metabolically that's now stable in the body. And if we go from alkyne, triple bond, to an alkene, double bond, and then an alkane, we can completely change the orientation across that bridge. So this would take you back, linear, so completely straight, planar, so this is where nice and flat, and there's a difference between a Z, this is a Z configured alkene, and an E configured alkene, and the more floppy alkane. So we can move from one to the next to the next. So this locks the structure in. It doesn't replicate the normal dihedral torsional angles of a disulfide, but what you'll see is that doesn't matter. You'll see in a minute. So we make these carbon-based manifolds. And in order to do that, we have to use some pretty sophisticated chemistry that won the Nobel Prize back in 2001, the catalyst, that is. And I spliced this in row just for you, and I think that you'll see why. So let me hit play. Hopefully I'll hit play properly. Here we go. So in the centre here, this is the catalyst. These are usually ruthenium-based catalysts. These are your monomers. And you can see what's going to happen here is it's like a partnership. They're dancing, and they're going to bump into each other, and they're going to form like a 2+2 cycloaddition, that's what we call it. When they bump, 4, and then they exchange partners.
That's just like my movie.
Exactly. So that's the end. So that's representative of a metathesis cycle. From 2 olefins, you come together to form that square. They exchange partners, a new olefin goes, and then the next one propagates into the cycle. Very similar to what I just saw with yours. So coming back to this, when we do dicarbo replacement, we can simplify the synthesis of some of these really quite treacherous structures to make because we use the metathesis chemistry to force various groups together, and it's the only way that they will couple. We can stabilise the protein in vivo because disulfide bridge is no longer being opened, can only be closed at any one time. And the information that we can get from doing those replacements tells us not only what the original disulfide might be doing in the native molecule, but it also can tell us about mechanism of action. And I'm here to tell you that most of what we know about the mechanism of action of hormones that we've been studying for more than 50 years is very incomplete. Very incomplete. So, why else do we care? This is just out of The Guardian, I think, of a couple of weeks ago, 4th of August. 5th of the medicines in Africa may be subpar or fake. This is very relevant to disulfide-containing peptides. They must, if you think of insulin, it must be kept at 4 degrees. It can't be above 4 degrees. It can't be frozen. And when that goes out of that very narrow band, it loses activity. And if you're a diabetic patient and you're getting a compromised medicine, you're in trouble. Likewise, you'll see a molecule later called oxytocin, which is a hormone that is very important for women after they give birth. So the moment the baby is pushed out, the vasculature must immediately shut down, otherwise the mother will bleed to death. And it is the leading cause of death in childbearing women, postpartum hemorrhage. And so most women in hospitals nowadays get jabbed in the bottom with oxytocin just to ensure that that vasculature shuts down really quickly. Okay, so 3 examples and then I'll be finished. So this is my favourite molecule. So we've been working on this hormone for about 15 years. It's a 51 amino acid peptide. Put that into perspective. Every time you put one amino acid, link it together. So you usually start off at one end and then you add the next and then add the next and add the next. It's 3 reactions. So 3 plus 3 plus 3 plus 3. You can see that that's a lot of work. And the first time we made this molecule, normally the hormone has 3 disulfides, you probably remember from undergraduate, 1, 2, and 1 in the A chain, in through here, disulfide in here. We wanted to replace this one with a carbon-based bridge. That took us an entire year to make. And at the end, we had 10 micrograms. So we had like a vial that we hoped had it in there. We could see the molecular ion. We couldn't see the molecule, obviously, 10 micrograms. And we gave it all to Adelaide and our testers, and the results came back saying that it's more potent than human insulin. So it told us for the first time that this disulfide was not critical for biological activity, although the literature said that it was. It's an invariant part of this structure. These disulfides, it doesn't matter if you're a guinea pig, a giraffe, a hedgehog, a human, a pig, whatever you might be, over 150 vertebrate species, your disulfide linkages are all in the same spot. Invariant. Absolutely critical in terms of the structure, but not important for its function. There are other parts of this molecule that are important too. Down here, this glycine, isoleucine, valine, invariant again. Why? Why is it that they don't change? Throughout the structure, there are various residues that have very modest changes. And sometimes, if you're a type 1 diabetic, some of these point mutations may have changed, but you have type 1 diabetes. And if you as an embryo have, changed out multiple of these or certain ones that are critical, then you won't survive. That embryo will not survive. So it's, nature makes use of this template and is very conservative about how it changes it, structure and for function. So we replaced this one. We had 10 micrograms. We needed to make some more. So we needed to change the synthesis. I'm not going to go into any of that, but changing the synthesis, we made a lot more of this molecule and we sent it over to Adelaide, and I deliberately flew over to get the results, and I was told the molecule was inactive. And I can tell you that was the longest Adelaide to Melbourne flight I've ever had, because I had to come back and say to the PhD student, who had then spent another year and a half perfecting the synthesis, that maybe we didn't get it right the first time and maybe the molecule was inactive. But as the story is always, there's always a happy ending, well, not always, but I'm going to call it, is that the first time we made the molecule, that bridge was in a cis conformation. When we changed the synthesis, the double bond was now in a trans conformation, and we could never check that because we'd sent the 10 micrograms to Adelaide in the first place to do the testing. So it turns out that control of the metabolic activity of insulin is exquisitely controlled by the conformation at that site. Exquisitely. On, off. On, off. So the disulfide is playing a very large role in that. And I can just go back to the previous slide and come back to this. Is that why that's important is that this disulfide up here in the native molecule controls how this helix sits in the active site. And so that GIV, which I said to you was invariant, is the thing that is unwinding here when it goes into the receptor. So it goes from being a cylinder, an alpha helix, to like a horn, like this. And as it unwinds, these residues move and it enables it to lock into the receptor. Without that change, it doesn't bind. So these little tiny things that we're beginning to understand with insulin are very important for its activity. So the last part of this story is we've been working on trying to replace this invariant disulfide bridge here, working on a hypothesis that the histidine binds to zinc. We already know that. That's the hexamer of insulin and how it stores in our body. But the zinc moves from here to up to this disulfide bridge, which causes this then to unwind and to lock into the helix. And we've been working on that for 10 years. Really tough problems, and what Ro just mentioned before, biting off these very hard projects, they kill you inside, but when you get there, very triumphant. Okay, so we're still working on that and the mechanism of action because it's important. Oxytocin, I mentioned this one before for postpartum hemorrhage, much simpler molecule, disulfide bridge in here. You can also take this out and make a carbon-based molecule. It makes it stable in vivo and it has a much longer half-life. When you make the alkyne version of this molecule, I'll show you what happens. Again, exquisitely controlled by conformational change, just 2 atoms taken out, but the hybridization locks it into place. So, had we read the literature, 2 oscillating stars here. So back in 1974, this paper was published. Now, had I read the literature, the rest of this story would not have come out. But in 1974, this paper was published saying that the mercury oxytocin derivatives, so this is where you go sulfur, mercury, sulfur, in putting mercury into that disulfide bridge, were found to have negligible biological activity in uterine contraction of a rat. Had I read this, I wouldn't have done this experiment. So this is absolutely false. So this is a uterine contraction assay in red. This is oxytocin. So how to measure this is concentration along the bottom, so low concentration here, and the amplitude of the contraction. So here's native hormone, here's mercurated oxytocin, higher amplitude, lower concentration. This has set the path of oxytocin research, also insulin, in the wrong direction for decades. It's quite clear that these hormones are regulated by metal binding through their disulfide bridge. Very important. And the last one, just 2 more slides. Pain, an unmet area of medical need for sure. If you've got chronic pain, the best that I can do for you is to give you morphine analogues. Addictive, and the major side effect, other than addiction, constipation. Really bad constipation. I'm not talking about just inconvenient constipation, I'm talking about hospitalisation constipation. So there's really not a lot that we can do for people with chronic pain, and the best that we can offer is something like gabapentin, which causes psychological distress, and this is shown down here in blue, and this is a measure of pain. So this is sort of a healthy individual, in pain individual being dosed with an analgesic and then coming back to normal. So here, if it returns up to the top here, for example, this animal is not feeling pain anymore. So this is the example of a cone snail, just out of something that you would pick up on a beach, and the cone snail makes this beautiful cocktail of thousands and thousands of molecules, and if you sequence those molecules, you can get to these very simple structures. They're about 16 amino acids together. They have interlocked disulfide bridges. And people fight over the mechanism of action of this molecule because it's a potent analgesic, as potent as a morphine analogue. So they fight. How does it work? Is it through nicotinic acetylcholine receptor inhibition? Is it through GABA B activity? You know, like, as a chemist, I don't care. But by making the dicarba derivatives of this, so using the synthesis, we replace this one and make this analogue, and then we take this loop and we make this one, and then we test them. And what we can clearly see is that the activity, the analgesic activity, resides in the first part of the molecule. So in fact, you only need to make a molecule this long or even this long, and even the G can be taken off. So just within that loop is a molecule that is highly, highly potent as an analgesic. So in through here, this is the red is the native conotoxin molecule. In purple is the mercurated, mercury S. And the reason why we make the mercurated analogues, I know everyone in the room's going, mercury, like what? You're not going to put it into a patient, but mercury S for chemists is a linear motif. And it gives us an instantaneous read on whether we should bother making the alkyne molecule, because the alkynes the catalysts are highly air sensitive and moisture sensitive. So you have to be really skilled in order to put an alkyne in. So making this molecule, 2 minutes to go. This is somebody in chronic pain. It's a rat. Chronic pain, you can see the firing of the nerve in through here, being dosed after 70 minutes. You can see it absolutely flatlining through here. And these little ticks here are electrical stimulation to show that the nerve is not actually dead. So this is showing you no pain being transmitted through this nerve. And so you can see that this return to almost pre-injury state up here with these cone snails. So there's a lot of work in this area that's being done in chemistry. A lot of focus goes into trying to work out what the bioactive motif is and how the flexibility of the molecule is important for its activity, and trying to lock it into that conformation. Really important. Finding out, locking it in. So my last slide here, a couple of slides. So I must say, I met Rosemary, I think back in 1996, thereabouts. We met on FRC and other panels where the dean at the time was silly enough to put us together in the same room. But it did actually dwindle over time, and I'd like to think that maybe that frequency of our, interaction was because we had more women in the Faculty of Science rather than thinking we can't put those two together. But it was a lot of fun, and we socialised together. We have a chaotic relationship. There's no doubt about that. Everything always seems to go crazy, but a lot of fun. And so I think that when you look at these photos, you can see 2 decades of camaraderie, and perhaps towards the end, a bit of a problem with alcohol, but.
That's where the naked men were.
Okay, all the best, Ro. Thanks.